Good afternoon, everybody. My name is Renske Vroom, and I am a PhD student at Radboud University in the Netherlands. I would like to start by showing you this picture of my city, Nijmegen. This was taken only yesterday. And you can see that the river that is passing the city has risen to very high levels and is now actually flooding the edges. And of course, now you may think, well, flooding happens all the time in the Netherlands. You have learned to live with it. But these events are quite extreme, and they are happening more and more frequently. These extreme weather events are not only happening in the Netherlands. We only have to think about the extreme droughts and fires in the Amazon and the Pantanal in Brazil to see that they are happening worldwide more and more frequently. Unfortunately, we as humans are responsible for these extreme weather events because we are drastically changing the climate. And this climate change is, of course, caused by our very high emissions of greenhouse gases. And one of the most important greenhouse gas sources is our food production. And because we now know this very well, we are looking for alternative sources of food and also sustainable sources of proteins. And one very important protein source worldwide is fish. Unfortunately, the problem with fishing wild fish is that overfishing causes problems with ecosystems and with biodiversity. And here we can see that a large amount of the world's waters is now being overfished. So to prevent this from happening, we have thought of another way to produce fish, which is aquaculture, or breeding fish in small shallow ponds. And because of this, aquaculture has boomed in recent decades. This graph shows the aquaculture production in Brazil, and we can see that it has increased over 100-fold since 1995. So this could be a very important way to farm fish sustainably. However, very little is known about the environmental impacts of these fish ponds. And among these environmental impacts is greenhouse gas emissions. And this brings us to the topic of my presentation, which is called Fishing for Methane. And I will show you the first insights into the variability, pathways, and potential drivers of methane emissions from fish ponds in Brazil. So why are we so concerned about these fish ponds as greenhouse gas emitters? Well, to optimize fish production in these ponds, we often put in a lot of feed and fertilizer, so the fish have enough to feed on. Along with the runoff that can come from the neighboring areas of these fish ponds, which are located most frequently low in the landscape. This causes the accumulation of carbon and nutrients, and the accumulation of sediment. And this carbon-rich sediment will break down and cause the depletion of oxygen in these ponds. So then we are left with an environment and a water column that is rather oxygen poor and rich in nutrients. And this is the perfect environment for the production of greenhouse gases, nitrous oxide and methane. So in my study, I focused on methane, which can be emitted through two different pathways. It can diffuse from the water column into the atmosphere, or it can accumulate in the sediment and form bubbles. And these bubbles can be released directly from the sediment to the atmosphere. So a few, not many, studies have been done on greenhouse gas emission from fish ponds. And most of these studies were done in China. And Yuan et al. Nicely reviewed these studies and found that the average emissions were about 105 milligrams of methane per square meter per day. We can see that this estimate is highly variable. This is the standard deviation. So as I said before, methane can also be emitted through the ablative pathway. However, only 2 out of these 19 studies quantified methane abolition. And it is estimated that methane abolition can actually contribute 42 up to 77% to total methane emissions. And this estimation is based on similar systems, which were mesocosms with or without fish. So this is just an estimation, and we can see also in this graph that the uncertainty range is still rather high. So it is very important to quantify both diffusive emissions and ablative emissions of methane from these ponds. So in our study, we wanted to make a first estimation of methane emissions from fish ponds in Brazil. We also wanted to estimate the contribution of abolition to the total methane emissions and quantify them. And last but not least, we wanted to identify potential emission drivers, because once we know these drivers, we can aim for a more sustainable way of agriculture with the lowest methane emissions. So last year, I was lucky enough to travel to Brazil and sample 52 fish ponds in 21 different fish farms. Most of them were located in the province of Rio de Janeiro, and 2 of them were located in the province of Minas Gerais. All of them were just north of Rio de Janeiro. So what we did, we measured diffusive methane fluxes with these floating chambers, and we measured them at the edge of the fish pond as well as the center of the fish pond. These floating chambers were attached to a greenhouse gas analyzer that measured the concentration of methane and CO2 every second. Then we also measured the ablative fluxes of methane, and we did this by installing these funnels, which were floating at the surface, and they were installed there for 24 hours. And all the bubbles were collected in these glass bottles where we could extract them and determine the concentration and the volume of methane. So I want to give you just a brief overview of the different fish ponds that we sampled, because they are all very different. Most of these fish ponds farmed tilapia, 22 farmed them on their own, and another 19 farmed tilapia mixed with other species. And these other species were mainly traira, lambari, and cara. And there were also some fish ponds that had wild fish species, and one of them had grass carp. We also saw a high variability in the sizes of these ponds, which could differ almost 2 orders of magnitude, as you can see here. This, for example, is a very tiny pond. Our barge barely fitted in there to measure the center fluxes, whereas this was an example of a very large pond that was also a natural one. So most of our ponds were excavated earthen ponds. One of them was excavated with a concrete layer, and 5 of them were natural ponds. The use of these ponds also varied a lot. For example, this pond was used for breeding, and they bred these fish in high densities in these hapas. Whereas in this fish ponds, they invited people to fish their own fish, and they could prepare them in this restaurant. So most of them were commercially used. Some of them were used for breeding and research. Others were used for private foods as a private food source, and some just for recreation. You could also see just by eye very big differences in trophic state. And this was often also related to the location of the ponds. So you can imagine that everything, that all the runoff that is collected here on this hill will eventually end up in the pond. And the same happened here. And here there was a cow farm just situated uphill here. So all the nutrients from this cow farm actually ended up in this pond and made it very eutrophic with floating macrophytes and high concentrations of blue algae. So coming to our first results, this graph shows the pond ID on the x-axis and the methane diffusion on the y-axis. And this methane diffusion was measured in the center of the ponds. The black dots represent the different replicates. So what we can see here is that methane emissions were generally quite low, with a median of about 11 milligrams of methane per square meter per day. And this is quite low also when we compare it to the values that were found by Yuan et al. In other papers, and to the IPCC emission factor for small ponds. Of course, here we have some higher emissions, but most of them were rather low. This graph shows similar results, but it includes the diffusive fluxes that were measured at the edges of the ponds in blue-green. And what was very obvious from here was that the emissions from the edges of the pond were on average twice as low as those from the center of the pond. And this indicates that it's very important when you measure methane emissions from a fish pond where you measure, and you have to take into account the spatial variability. And this variability is probably caused by an uneven distribution of sediment within the pond, because they're usually a little bit deeper in the middle than at the edge. So the sediment accumulates there. So this graph shows again the pond ID on the x-axis and the emission on the y-axis. But here we can also see the ablative fluxes in purple. These were measured only in the center of the pond, and the yellow bars are again the diffusion in the center. And what is very obvious from here is that ablative fluxes could be very high. Here they're almost 600 milligrams per square meters per day, much higher than the diffusive fluxes. So ebullition is a very important pathway for methane in these systems. And the contribution to the total flux could be even up to 96%. The median of this contribution was 63%, which coincided quite well with the estimation made in a paper by Kosten et al. A little side note is that these values may be even underestimated, because the method we used to determine the methane concentration in these bubbles was actually unable to measure such high concentrations as were present in those bubbles. So this is the lower estimate of the concentration, the bubble concentration. Just again for comparison, the mean ablative flux was 69 milligrams of methane per square meter per day, which was still lower than the emissions found by Yuan et al., but definitely higher than the IPCC emission factor for shallow ponds, small ponds. So lastly, we are still a little bit working on this, but we are trying to unravel the drivers behind these variable fluxes. And this is a PCA plot that shows the diffusive fluxes of methane and the methane concentrations in the water layer. And it also shows all the different environmental variables that we measured. So what we can see from here is that diffusive fluxes and methane concentrations in the water table coincided quite well. So when one is lacking the equipment to measure diffusion directly, water concentrations are quite a good indicator. And these were negatively linked to oxygen concentrations, blue algae, brown algae, and phosphate concentrations in the water, and also to pH at the water surface. So in conclusion, we saw that diffusive fluxes from fish ponds in Brazil were rather low relatively, but highly variable. And there was also spatial variability within ponds with lower emissions at the edges than in the center of the ponds. And we think that we could explain these low methane fluxes by quite high oxygen concentrations in the water layer. The most important thing that we found out with this study was that ebullition had a very high contribution to total emissions, up to 96%. And this finding actually has huge implications for emission estimates, such as the emission factor that is determined by the IPCC. And to really know the extent of these emissions, it's really necessary to measure them on a longer term, for example, periodically during a year, because these emissions are very episodical and can depend, for example, on air pressure and weather conditions. It will also be very interesting to unravel the influence of fish species and pond management on these emissions, because of course the density of your fish, their life stage, and the species will determine their behavior and can also affect these emissions directly. I would like to end with the take-home message that low-emission fish farming is possible, because we did see some ponds that had both very low diffusive and ebullitive fluxes. And with the right management strategies, I believe that low methane emission fish farming is possible, and that we can create a protein source very sustainably in this way. So with that, I would like to finish, and I would like to thank all the people that helped me during the fieldwork and the lab work, and it made me feel really at home in Brazil. I had a really great time. And I would be glad to take any questions that you have, either during the session or by email. This is my email address. And I would like to thank you very, very much for your attention.